So, friends, after getting introduced to ANOVA and F test, now in this lecture we will get an introduction to design of experiment. So, let us first start with the introduction. What an experiment is actually. So, an experiment is usually planned and can be described in the following steps. So, whenever we are planning an experiment, we will consider these many steps. By knowingly or unknowingly, we will always follow these 6 steps. First of all, you will get the introduction of problem. What problem? For what you want to design an experiment. So, first of all, you have to get introduced to the problem. Then, after getting idea of the problem, you have to tell the hypothesis, whether H0, HA, how we are stating hypothesis, we have already studied about that. Then after stating hypothesis, you have to describe the experimental design. Here in this area, this design of experiment will work. So we have to describe the experimental design. After describing the experimental design, you have to perform experiment and you have to collect the data from the experiment. Now after collection of data, you have to analyze the data resulting from that particular experiment, and after analysis you will interpret how your results gone and what your results is indicating. So this is how we are planning any experiment. So these all the things we have to keep in mind when we are planning our experiment. So now what is design of experiment? So the advanced planning of experiment, you have to note it here, that advanced planning of experiment to achieve objective relevant to the problem. So what problem you have identified, in order to get relevant result or relevant solution to that problem, for that you have to plan your experiment in advance, and such planning is called as design of experiment. Such planning of experiment is called as design of experiment. And in that design of experiment, we generally specify what we specify in that design. So first is the treatment. So what all treatment you are going to test? Treatment means what are the factors? Suppose you are working on you are having 3 kind of feeds. You are having 3 kind of feeds: feed 1, feed 2 and feed 3. Now you want to know which feed is more effective in increasing milk production. So that means you are having 3 treatment: feed 1, feed 2 and feed 3. So this treatment you are in design of experiment, you have to identify your treatments. Then the experimental unit, on what you want to apply the treatment. So experimental unit, if you want to increase the milk production, that means your experimental unit will be the animals, lactating animals. So those lactating animals you have to identify. So you, in a design of experiment, you are specifying the treatments, you are specifying the experimental unit, that is the experimental animals, and the rules by which the treatments are allotted to experimental unit. So you will have 2 things. One is treatment, another is experimental unit or experimental material, and third one is the rule by which you will allocate the treatment to that experimental material or experimental unit. And after that, you will get the data, you will measure the data, and you will record the data that you will get after applying treatment on experimental material. So this is basically the design of experiment. Now, what are the different associated terms with the design of experiment? So many of the terms we will discuss those terms when I will be actually discussing about various design of experiment, but few terms that will come everywhere, or means whatever I will discuss in design of experiment, you will get these terms always. So it is better to learn about these terms in advance. So first is experiment. What is experiment? So what is experiment? I have already discussed just few slides back. It is a planned inquiry. So we are inquiring about something, but that inquiry is planned. How it is planned? A planned inquiry to obtain new fact. For what? We are planning inquiry for what? To obtain a new fact. New fact means, suppose you are having 3 feeds and you want to test which feed is effective in increasing milk production. That means you are establishing a fact that feed X is increasing the milk production. So for that you need a planned inquiry. So experiment is a planned inquiry to obtain new facts or to confirm or deny the result of previous experiment. It can also be there that many a times you will be asked to test whether a particular firm or particular company or particular material is said to have effect on milk production. You do an experiment and confirm or deny. So for confirmation or denial also you need to plan an experiment. You need to have a planned inquiry. So those planned inquiry which is done to obtain either new fact or to confirm or deny a claim, such planned inquiry is called as experiment. Okay, why we are doing this? Because so that we can recommend a variety. Variety means many in plant experiments. I have already told you that this design of experiment, ANOVA, etc. has been developed initially in the plants. So many a times plant example will come. I will try to give example from animal science, but still the plant examples will come also. So variety, many means suppose a scientist is working on wheat and he found that a particular variety of wheat is yielding more. So for confirming this claim or for making this claim, he need to do an experiment. So this experiment or planned inquiry is help us in recommending a variety or a particular procedure or a treatment. Okay, suppose you have discovered a new drug. How you will tell the world that this drug is effective in curing a particular disease? For that you have to do a planned inquiry or an experiment. Now the second one is experimental design. Experimental design. So what is experimental design? It is a specification about planning and conducting experiments so that more precise and unbiased estimate of treatment difference are obtained. So what is experimental design? Suppose you are having 3 treatments or 4 treatments or number of treatments. Number of treatments means this feed one, you are having group of laboratory animals and now you want to test your 3 or 4 feeds. 3 or 4 feeds that you have developed, F1, F2, F3, F4. Now you want to tell that which feed is better. So for that you need to allocate those feed to the group of animals. How you will allocate those feeds? So this specification about planning and conducting of experiment will come from experimental design. This experimental design will describe how you will group the animals and how you will apply treatment to those group of animals. So that is called as experimental design. Now the another term is treatment. The treatment we are discussing. So the treatment, it refers to constant or condition. What it refers? It refers to constant or condition. Treatment can be anything. Either a feed is a treatment, or suppose you don't want to give water to a particular animal, then water is a treatment. Water will be a treatment. Animals with water, animal without water. Or if you want to give different doses of water to animals, so those doses will be a treatment. Or suppose you develop few drugs, 3 or 4 type of drugs. These 3 and 4 type of drugs will be treatment. Or in case of plants, if chemical that we are applying on plants for removing or removal of any disease, pesticides, different pesticides, that pesticide will be a treatment. So treatment can be anything. Suppose you develop a new method for estimation of something. So 2-3 methods you have developed. So these methods will be the treatment. So what treatment refers? It refers to the constant or condition applied to an experimental unit. So whatever treatment, whatever constant or condition, these treatments are condition that you are applying to experimental unit. It is a class, group or category under comparison. Example, variety of feed, we have already discussed. Medicine you have developed or drug you have developed. Application of drugs, etc. Application of drugs means the amount of drug, different concentration of drugs. So these are all coming under the treatment. So these terms you have to remember: what is experiment, what is experimental design, and what is treatment. Another term related to this is experimental units. What is experimental units? So as of now you might be in position to tell what are the experimental units. So this is the smallest unit of experimental material to which the treatment is applied. Suppose you are having 100 animals, group of 100 animals. You are having group of 100 animals. Now you form within group, you form two groups. First is group of 50 animals and another is another group of 50 animals. Now in each of the animal, on each of the animal, you are applying a particular drug you have developed. So on each animal, that means in a group there are 50 animals, on each of the 50 animals you are applying drug 1. So the smallest unit on which you are applying a drug is called as experimental unit, and that smallest unit is animal. So that animal is your experimental unit. Suppose in case of agriculture, suppose this is the plot, this is a plot, or this is a field. So you divided this field into different groups. So here suppose you are applying one chemical, one pesticide P1, here P2, and here P3. So these smallest unit on which you are applying a treatment are called as experimental unit. So I hope this experimental unit concept is clear to you. For better understanding of design of experiment, you need to get clear-cut understanding of these terms. Otherwise, it will be difficult for you to understand the other advanced concept of design of experiment. Now another one is experimental error. The failure. What is experimental error? So you just try to understand the failure of two identically treated experimental unit to yield identical result. Suppose you are having two animals. Suppose this is dog 1 and this is dog 2. You applied same quantity of drug T1 on D1, and here also you applied same quantity of drug T1 on D2. Even then, a particular disease that D1 was suffering, same disease was suffered by D2 also. But after application of T1, D1 get cured, but D2 did not get cured. D1 get cured, but D2 did not get cured. So what may be the reason? The reason is unknown, because you applied same treatment, and the dog was of same age, sex, breed, and disease was also same. So what went wrong? There may be something which is causing the difference, and that something is called as experimental error. So now you just try to understand the definition of experimental error. The failure of two identically treated experimental unit to yield identical result is called as experimental error. So this error will cause the failure of repetition of the result. So that is called as experimental error. Now another term is layout of design. So what is layout? As we are planning a house, construction of a house, we are preparing a layout. Similarly, when we are planning an experiment, we prepare a layout for what? Layout here in case of experimental design, layout we are preparing for application of treatment. So what layout is telling? Layout of design, it is a placement of treatments to experimental site that may be over place, time, or type of material. So how we can understand it by this? Suppose this is the 3 animal. So three animals or uh, three animal. So we decided to put T1 on first animal, T2 on second animal, and T3 on third animal. So this layout we have prepared that on first animal I will prepare T1, second animal T2, and third animal T3. In another way you can understand it by this. Suppose you are having one animal, you give a particular feed F1 in summer season, and you recorded the milk yield. You recorded the milk yield. Then you give F2 or F1 again same treatment in winter season, and you recorded the milk yield. Then again same treatment, same feed on rainy season and recorded the milk yield. So this treatment is being applied over different period of time. So that is also called as layout of design. So before conducting an experiment, before conducting an experiment, in design of experiment, you have to prepare your layout. Why it is called as layout? Because in agriculture, a field is, suppose this field is being divided into different parts, these parts. So now they planned that this T1 will be applied here in first unit, then third, again in third unit T1 will be applied, then in here also they will apply T1, here also they will apply T1, then next one they will apply T2, then T2. So the rule by which they are applying different treatments into different blocks is called as layout. So layout they have to explain before designing an experiment. Now the next now what are the purpose of design of experiments? So by this you have already understood what is the purpose of design of experiment. Just I will be reading it here. The first purpose is to obtain valid conclusion. So valid, when you are suppose how to determine that your conclusion is valid or not. So for that a standard methodology should have been developed, and that standard methodology is design of experiment. Otherwise anyone will do a small experiment or experiment which is not standard, and he will tell, or even he will not do an experiment and he will tell that okay, this particular drug is very effective in increasing milk production. So how people will test whether his claim is right or wrong? For that you have to design the experiment. So to obtain valid conclusion about treatment effect in controlled condition by getting unbiased effect. So for getting unbiased effect of treatment under comparison. Second, to get estimate of experimental error. You know, by application of treatment on same animal, on same period, we are not getting the results. We are not in a position to repeat the result. There are some discrepancy in the result. That discrepancy is called as error. But our aim should be that to minimize the error. If a particular thing or particular methodology is in a position to minimize those errors, that we will consider when designing an experiment. Okay, to get the estimate of experimental error or variation caused by uncontrolled sources of variation. So for that I will this uncontrolled sources of variation I will be discussing in the later when I will discuss about the principles of design of experiment. Another is for objective generalization of experimental result. So we have to plan our experiment in such a way that suppose today here at this place I conducted a result and got result X, and on very next day somebody is performing experiment and he is getting result Y. So this why there should be such kind of discrepancy? Because the design of experiment was not standard, it was not right. So to minimize such discrepancy, or that means to make the experimental result generalized, whenever, whoever, at any time, if he is doing experiment, he should get same kind of result. Such this is the purpose for design of experiment. Then next is the principles of design of experiment. So what are the principles of design of experiment? So there are three basic principles of design of an experiment. All these principles help in reducing the experimental error. So purpose of design of principle of design of experiment is to reduce the error. So we will see in next few lectures that how this error of the experiment is very important, or experimental error is very important. So many a time we want to reduce the experimental error. For that we have to apply the design of experiment, principles of design of experiment. So three principles are replication, randomization and local control. Replication, randomization and local control. These are the three basic principles of design of experiment. So we will see each one of them one by one. So first of all, replication. The repetition of basic treatment. Replication means repetition of basic treatment. Basic treatments means suppose you are having suppose you are having three groups of animal, 1 2 4, then 1 2 3 4, then 1 2 3 4. So you are having three groups of 4 animal each. Now you want to apply the treatment. Suppose you have developed one drug and now you want to apply those drug on this animal. So if you are putting drug 1 only on this animal, then you are putting drug only on this animal, and suppose this animal and this animal is getting treated. Suppose you applied the drug 1 on animal 1 and that animal got treated, but on next day when you are applying same drug D1 on animal 2 and this animal is not getting treated. Again on third day you applied D1 on third animal and this animal is again getting treated. So 2 times it is getting treated and 1 time it is not getting treated. So it was decided that there are many factors within the animal which is causing deviation in the result. So for that, to avoid that deviation, it was thought that if we are repeating the particular treatment on many animals, so sometime we will get an average result, which is the property of that particular drug, and the deviation that is caused due to the variation in animal will get cancelled out. The variation in the animal, the deviation that we are getting result due to the variation in the animal, that will get cancelled if we are repeating the treatment on large number of animals. So that is the basic concept behind the repetition of the treatment. So replication is the repetition of basic treatment or treatment combination under study is called replication. So replication is the repetition of basic treatment. Why? In order to avoid error, so that error get cancelled out. So here, if single treatment, suppose there is single treatment or treatment combination is applied R times means 1 2 3 up to R times, then we say that there are R replication. So for any design of experiment, the basic principle is of design of experiment is the replication, that is repetition of basic treatment. So this should always be there. If you are applying treatment, the application of treatment should be replicated so that error can be minimized. So what are the advantages of replication? The increase in replication increases the precision. We have already discussed how it is increasing the precision by reducing the experimental error. Why it is increasing precision? By reducing the experimental error. And this increase in precision can be observed by two things. First, if you are increasing the replication, then there will be reduction in standard error. So standard error and replication are inversely proportional. So if you are increasing the number of replication, standard error will be reduced and thereby precision will increase. Another factor will be there, that precision increases with t distribution at various degree of freedom. So t distribution, precision increased with t distribution at various degree of freedom. So you see here, degree of freedom is inversely proportional to table value of t. So if you increase the degree of freedom, the tabulated value of t will reduce. That means the experiment will be highly sensitive. Small changes can also be detected. So that is why we are replicating the treatment. And another one is that degree of freedom is also inversely proportional to confidence interval. So if degree of freedom will increase, how degree of freedom will increase? If we are increasing the number of replication. So if degree of freedom will increase, confidence interval will be reduced and thereby the experiment will be more sensitive and small changes can also be detected. There will be reduction in experimental error. So this is how the replication of treatment works for the betterment of design of experiment. Now second is, the replication provide us the experimental error. If we are not applying replication, that means only one treatment, you are applying a treatment at one time. So whatever result you will get, that we will think that okay, this is the effect. But when we are applying replication, you will get the value of error also. So it is giving the experimental error value. If we are not replicating our treatment, we will not get error. So this error is very necessary for comparison of result. So with which treatment you are getting lesser error. So this error is the basis of comparison of result. Then second is the replication. It is must for consistency and accuracy of the result. Consistency how? That means our objective should be at whatever times we are applying our treatment, result should be similar. That means your treatment effect should be consistent. If it is not consistent, that means there is some problem in the treatment. Okay, and it will, in this way it will also increase the accuracy of the result. Then it makes the test of significance possible. Why it is making test of significance possible? Because of the because due to replication of the treatment you are getting the experimental error. Otherwise, if there is no replication, there will not be error and there will not be in a position to test the significance. It provides more error degree of freedom. Okay, we have already discussed. It provides more error degree of freedom. Hence, test of significance become more sensitive. That we have already discussed. Smaller difference between the treatments are also detected. So these are the advantages of replication. So how replication is a basic principle, how it is working in reducing error of the experiment or increasing consistency and accuracy of the result. The next principle is randomization. What is randomization? Randomization of what? Randomization in allocation of treatment. So main thing is treatment. So you in experiment you are seeing the effect of treatment. So how these treatments are being allocated to different experimental unit? You know what I mean by experimental unit. The allocation of treatment to different experimental unit by a random process is known as a randomization of treatment. So application of treatment to different experimental units. Suppose you are having 10 animal and you are having two treatment T1 and T2. So which animal should be given with T1 and which animal should be applied with T2? So if we are doing it by random process, then it is called as randomization. Otherwise, if we are doing by certain biasness, that okay, this animal A1 is looking good, so we will apply it on it you will apply T2. Animal 2 is looking weak, so we will apply T1. So in this, if we are using, or if you are keeping some biasness in application of treatment, our whole experiment will be failed. So that is why the second principle of design of experiment is the randomization. Randomization in what? Randomization in the allocation of treatment to different experimental units by a random process. So this randomization refers to allocation of treatment to various experimental units such that each treatment has an equal chance. So this the process randomization, it should be random. What is random? We have already seen random when we were discussing about sampling design. So in that, random means each and every treatment should have equal chance to be applied on each and every animal. So that is called as randomization. So each treatment has an equal chance of being allocated to the experimental unit. It is done by random number table. So we have already discussed there when we were discussing about sampling design about the random number table. So in Excel sheet, if you will apply syntax is equal to rand between. So with this function you can generate the random number table, and based on random number of table you can apply the treatments. So this is about the randomization. So what are the advantages for doing randomization in allocation of treatment? So randomization helps in getting unbiased treatment effect by ensuring unbiased estimate of error. So only our motto for randomization is to avoid biasness, and if you are avoiding biasness, you will get the unbiased estimate of error. So getting unbiased estimate of error is very important for getting accurate result. It provides the basis for objective interpretation of the experimental result. So when you are unbiased in application of treatment, you will get the objective interpretation of result. Otherwise, your result will be tilted towards one side. It will be biased. Then this also forms the basis of test of significance. So your test of significance will be valid if your allocation of treatment is randomized. Otherwise, there will not be any meaning of doing experiment. The fourth one, it ensures the independence of observation. So if your treatment is independently applied or randomly applied, the result observation will be also independent, which is necessary for drawing valid inference. Otherwise, your inference or interpretation will not be valid. It ensures equal chance for every treatment. If you are allocating treatments randomly, then we know in random allocation of treatment, random is each and every treatment have equal chance of being applied on each of the experimental unit. So it ensures, randomization ensures equal chance for every treatment being allocated to each of the experimental unit in addition to independence of errors, and it also makes the test of significance valid. So now we have completed two basic principles of design of experiment. First one is replication and second one is randomization. Now the third basic principle is local control. We are not applying the third basic principle, local control, on each and every experiment. So when we are applying local control, that we will discuss in the next few classes. When it will be necessary, I will be telling there, in this condition local control is applied, and in another condition only these two. But the thing is that the first two principles, that is replication and randomization, is being applied universally on each and every design of experiment. So what is local control? It is the procedure to control inherent or natural variation. This is a procedure to control inherent or natural variation present in experimental material. It's called as local control. So many times in your experimental unit, experimental material, there will be variation. But when there is variation in experimental material itself, even after applying treatment randomly or replicating it, you will not get the error free result. So for that, local control is being applied. The procedure to control inherent or natural variation present in the experimental material. I will give you one example, or just see one example. Suppose this is a field. This is a field of paddy. Suppose this is the field of paddy. You can note it here. Here in this side, a river is flowing. River is flowing from nearby this field. Now you see, you want to test two fertilizer 1 and fertilizer 2. For that, you divided your experimental unit into two parts. So what you will do, in first part you applied F1, and in second part you applied F2. So now you can see, or you are knowing that by this side of the field, a river is flowing. So due to the effect of river, the fertility of this portion of field will be increased, whereas it will not be that much in the second part of the field. So the application of fertilizer on this side means the side in which the river is flowing, this side will be more, the effect will be more. Even if your fertilizer is not having that much merit, even then you will get better result on that side, or you will get better production on that side. So in this way you can see that there is inherent gradiently fertility will be reducing from this side to this side. That fertility of the soil will be higher in this side and it will be lower in this side. So in order to control this inherent problem or inherent variation in the land, what we will do, we will apply local control. So how we will apply local control? Just see, suppose this is the field and here the river is flowing. So what I will do, I will divide the experimental material, that means field, into these three blocks, and I will apply the treatment like this: F1, F2 or F3, or F1, F2, F3. So by this way, each of the fertilizer will get similar kind of land. Thereby the natural variation present in the land will not affect the result, that is production. So by this way, this is the method of application of local control. So the principle of making use of greater homogeneity. So by application of local control, we are ensuring greater homogeneity in the groups of experimental unit for reducing the experimental error, and this is called as local control. So this is done by dividing the experimental material into, so these are the two groups. So between group, and the variation present in this unit only is called as within group error. So there will be two type of variation: between group variation and within group variation. This is about the local control. Now the between group error should be as large as possible. Between group, that means between group we are applying different kind of treatments, so it should be as large as possible, while within group error should be as small as possible, because within group error we don't want within group error. That is why the local control is being applied. So this was about the local control. So now the three basic principles of design of experiment we have already covered. The first one was replication, second randomization, and third was the local control. Now Another uh, another another control is the statistical control. This is not coming under the basic principle of design of experiment, but additional information I have used here. The use of additional information, example on pre-experimental condition. So before experiment you are doing something for making the experimental material uniform. To reduce experimental error is known as statistical control. So whatever things you are applying before experiment to make the experimental material uniform is called as statistical control. Taking or recording the information on one or more character associated with the character under study. Suppose you are studying about the milk production, but for safety you have also taken some measurement on other traits which is associated with milk production, and you are analyzing the experimental error with the analysis of covariance. That is, you will come to know in the part of the lecture, or sorry, in the later classes, analysis of covariance. So this analysis of covariance is helping in the reduction of experimental error, and that is called as statistical control. That means with a certain statistical method you are controlling the error. So that is statistical control. So with this I come to the end of our introduction class on this design of experiment. Here in this class we have discussed about the various terms associated with the design of experiment and the principles of design of experiment. So in next class we will see the actual design of experiment, what are the statistical method by which we are doing the design of experiment. So thank you.